Okay, so Section B of 2017 DEB mock. Alright, so Section B are your experiment questions. By now, hopefully you all know how to do your experiments upside down, inside out, back to front, and in 20 different languages. Alright, so for question 7, it says, in relation to scientific method, explain each of the following words. So what is a hypothesis? A hypothesis is a suggested explanation for an observation. It's, in other words, it's an educated guess. Alright, a replicate is a repeat of an experiment, and you have to learn off the word for word definitions for both of those. Alright, then part B, it says to demonstrate osmosis, 2 cellophane bags were placed, half filled in a 3% salt solution. The bags were placed in 2 beakers, one containing 10% salt and one containing water. Both were left for 30 minutes. Describe using words or a diagram the appearance of the cellophane bag. Alright, so in the 10% salt solution, the cellophane bag should have shriveled up because you had less water outside the bag than inside the bag, so the water would have left the bag, so the cellophane bag would have shriveled. In the distilled water, you have more water concentration outside than inside, so the cellophane bag would have taken in water, so it would have swelled up. And you could have explained it or drawn it. Answer the questions in relation to an investigation you carried out to show the effect of an environmental factor on the rate of photosynthesis. So this is your pondweed experiment. The easiest environmental factor to discuss is light, and how you discuss that is you say you moved a lamp or you moved a light towards or away from your plant. Right? How did you measure the rate of photosynthesis? You counted the number of bubbles produced per minute from the cut end of the Alida stem per minute. Okay, and you have to stress that you counted the bubbles for a particular amount of time. Okay, if possible, also say you got the average. Then it says answer the question in relation to investigation you carried out to determine the conditions required for germination of seeds. So what were the conditions necessary? You need oxygen, you need moisture or water, and you need a suitable or adequate temperature. Right, to describe the control you used. So the control you used was seeds given all 3 conditions, so you had seeds that grew. And in the course of your practical work, you used iodine solution, give 2 different sources or uses for iodine solution. Use it to test for starch, as in if you add iodine to starch, it goes blue-black. And you used it to stain your plant cells. It stained the starch in your plant. Question 8, it said the human circulatory system is described as a closed system. So what does that mean? Closed means that the blood is contained within a continuous system of vessels at all times, or your blood flows through your arteries, your capillaries, and your veins and your heart at all times. It doesn't pool anywhere. Distinguish between pulmonary circuit and systemic circuit. So it wants to distinguish, so they want 2 sentences, 1 on each. So your pulmonary circuit is the circuit from your heart to your lungs and back to your heart, and systemic is from your heart to the rest of your body and back to the heart. So for both of those, remember to state that it goes back to the heart. Right, it asks you about your dissection then. So it asks you, how did you distinguish between your ventral and your dorsal surfaces of your heart? In other words, how did you distinguish between the front and the back of your heart? The ventral surface was more rounded. You've got thick-walled arteries on your ventral side. The ventricle on your left-hand side of your heart was thicker. It was firmer than on your right. So basically, how I would answer that, as I say, I pinched the heart and I worked out left and right side. It's the most simplistic answer. There are 4 main blood vessels attached to your heart. How did you identify which were arteries and which were your veins? Arteries have thick walls, they have no valves, and they are linked to the top chambers of your heart, in other words, your atrium. Veins have thinner walls, they have valves, and they are linked to the lower chambers of your heart, your ventricles. In terms of dissecting your heart, normally, the only tubes you see are the veins, because the atriums have been chopped by the butcher. Right, then it says, describe how you dissected the right side of your heart to enable you to identify the internal structures. Right, so in terms of the description, they want 4 sentences. So you place the heart, ventral side, up in your dissecting tray. You used a scalpel. You made a cut and it's saying one vertical and one horizontal in the right ventricle to view your tendons, your papillary muscle, and the flaps of the tricuspid. All right. In terms of what I would say you placed your heart on the dissecting tray, you used a scalpel, and then you cut up the heart. Okay, so you cut up the heart, peeling it back, looking for your valves. All right, so on the right side of your heart, you are looking for your tricuspid valves. And you could also then say that you observed the thin wall. Okay, and then you identified your arteries and your veins. Okay, outline how you located the semilunar valves. Your semilunar valves, you cut down the length of the aorta, or you cut down the length of your pulmonary artery. Normally when you're looking for the semilunar, you go with the aorta because you will find the entrance to the coronary arteries there. 2. Right, question 9, it says, are fungi prokaryotic or eukaryotic? They're eukaryotic, meaning they have membrane enclosed nucleus and mitochondria. Right, so give a reason, I've just answered it. They have a membrane bound nucleus, they have mitochondria, and they can be multicellular as well. Right, answer the following questions in relation to the growth of leaf yeast in the lab. So the sterile nutrient medium should be used. What's the significance of it being sterile? Sterile means it is free from all microorganisms. So in other words, there is no contamination. It will be a fair test. Suggest one reason why leaf yeasts are more plentiful in July than in March. There's more leaves on your trees, it's warmer outside, there's less rain, so there's better conditions for growth of the leaf yeast. How did you introduce the leaf samples into the sterile medium? So according to them, you used a flamed sterilized tweezers, and you attached the leaf yeast to the lid of the petri dish using petroleum jelly, so Vaseline, and you attach it with the upper surface of the leaf into the Vaseline. So the lower surface of the leaf is facing your agar. How did you incubate the petri dishes once you'd attached them? You incubated them for 3 days, and after your 3 days you inverted them or turned them over, and that's about it really. Alright, how did you recognize the presence of your yeast? You had pink colonies on the agar formed opposite the leaf, and use axis, draw a graph to show the number of leaf yeasts varied. So when you have it, it looks like your bacterial curve. So you have a lag phase where the leaf yeast is basically sitting there adapting to its environment. Then you have the log phase where it reproduces like mad, the stationary phase due to the fact that toxins have built up, and then a decline. Okay, so that would be your section B.